Hello everyone, I am Asmita Bharti, pursuing my PhD from Indira Gandhi National Open University, New Delhi, India. First of all, I would like to thank you BSI for selecting me and giving me this opportunity to present my work here. So, introducing to my title, identifying potential metabolic protein and their crosstalk in neurodegenerative disorders via genomic and proteomic approach. So, this is the introduction, small introduction to my work. Neurodegeneration is a pathological condition involving progressive loss of neuronal cell in the brain region. These conditions usually develops and symptoms tends to appear later in life. The most frequent and the most prominent neurological disorders are Alzheimer that is affecting around 50 million people, and Parkinson is the second most and it is affecting about 10 million people globally. So my approach is in silico approach, that is involving the high throughput RNA sequence data for genome analysis and mass spectrometry data, that is MSMS data for proteome analysis. And both of the approaches has been done by using primary data set, and these were used to find out the metabolic protein as well as the associated genes involved in neurodegeneration. Differentially expressed genes and their crosstalks will help in revealing possible molecular pathophysiological pathways at molecular level that can aid in the early detection, protection or prevention of neurological disorders. So first of all, looking into the Alzheimer disease here in this figure, it can be seen that in the normal brain, in a healthy brain, the neuron are connecting with each other smoothly, that there is no hindrance, nothing at all. And in the Alzheimer disease brain, there are abundance of proteins like tangles are forming inside the neurons environment, and synapses loss and cytokinin over accumulation, overproduction, leading to the formation of amyloid beta protein in the brain cells. So ultimately they are hindering the connection between one neuron to another neuron. So coming to the symptoms, here we can see that in few symptoms I have mentioned here: impaired learning, memory loss, language problems, sundowning, etc. Causes could be the deintegration of amyloid beta protein extracellularly called plaque, and the accumulation of tau protein intracellularly called tangles. Preventions are few medications and lifestyle like sleep cycle, food habits, exercise, smoking, etc. So now coming to the Parkinson disease, here we can see in the normal brain cell, synapses is happening smoothly and lots of chemical signals are there, and they are moving one neuron to another one. And in the disease case, very few signals are there, and there are no communication between one neuron to another neuron. And similarly as Alzheimer, I have mentioned few of the symptoms in for of Parkinson, that is tremors, body balance difficulties, dementia on later stage of the disease, and sleep abnormality is majorly seen in the patient. Causes could be abnormal accumulation of alpha synuclein, environmental toxicants. Prevention could be medications, and low protein diet is suggested to the patients, and lots of water to avoid constipation. So now coming to the method. So for RNA sequence high throughput data analysis, my steps are starting with the downloading of publicly available data, and I have downloaded it from European Nucleotide Archive. For Alzheimer, it have 8 control and 9 case samples, and in Parkinson dataset, it have 23 control and 19 case samples. After successfully downloading all the datasets, were trimmed, and the quality check has been done using fast QC tool. After checking the quality of the data, I moved on to alignment. So for aligning my datasets to human genome star tool, which is a splice transcript alignment or to a reference, is basically a liner tool for RNA sequence data. It basically map the given data set to the library, and you can create your own library and you can use the already available library as well. But here in my research, I have created my library all by myself by using the genome generator of the start tour only. So after aligning all the data sets from Alzheimer and Parkinson, feature counting has been done, and feature counting has been done by using a Python package. It basically take the annotated file and align the output file of the star tool. Either it is a BAM format or it would be a SAM format. So after feature counting, differential expression of the gene has been observed, and for that DSEC2, which is a bioconductor package, has been used, and for that RStudio was the platform where I have done the differential gene expression analysis. After doing the differential gene expression analysis, regulation of gene was studied, and after checking the upregulation and downregulation of the genes, I move forward with the upregulated genes, and using the upregulated gene set, I identified the protein-protein interaction and top 10 hub genes. And along with that, I have checked the gene ontology also. And the another approach is proteomics approach, which is a for proteomics approach. I have used the mass spectrometry high throughput data analysis, and this analysis data is similarly downloaded from the PRIDE database. PRIDE archive. In Alzheimer there were 16 samples, and in Parkinson there were 10 samples. For mapping the raw data, MaxQuant tool was used, and it's a standalone tool for proteomic data search. And the parameters was mixed cleavage 2 was set to 2, mass tolerance was set to 20 ppm, fragment ion was 0.5, modifications that are fixed are cysteine, and variable modifications are deamination at asparagine, oxidation of methionine, and acetylation of lysine. After running MaxQuant successfully, I got two text file as an output file. One file contains the protein group, another contains the peptide groups, but I was more interested in the protein. That's why I took the protein group and removed the contamination from that file, and find out the common protein that are present in Alzheimer and Parkinson as well. So using that common protein, I identified the protein interaction and did the enrichment analysis, and as well as the top 10 hub protein, and check the interaction with other protein. So here is my result. So RNA sequence analysis has been done successfully, and after doing all the steps previously described, which has been described in previous slide. From Alzheimer, the gene counts were 41293, and for Parkinson, gene counts were 43498. And so far I can see, it can be seen here that 91.2% of the gene were found common for both the disorders, Alzheimer as well as Parkinson. But after applying filters or taking out the upregulation genes and downregulation gene, only 73 genes are common in Alzheimer and Parkinson that are upregulated, and rest of the 28.3% of the genes were downregulated, and that are common for both Alzheimer and Parkinson. In both of the cases, 88.2% of genes were downregulated. So coming to the result, RNA sequence data analysis lead to the prediction of protein-protein interaction. So for that, 73 common differentially expressed genes were found to be upregulated, and there exist 64 numbers of nodes, which is a gene, and 464 interactions, which are edges. And filters at the system level, organ level has been applied here, and that organ are the nervous system and the muscles. So only this much of genes were left in systems such as nervous system and muscles. Out of 73 common genes of Alzheimer and Parkinson that are differentially expressed, have several perspectives, and I have considered it into look into hub genes and identification of clusters of genes. So in figure number 7, interaction of differentially expressed genes which are commonly expressed in AD and PD, highlighting hub genes. The boxes is representing the gene which is a node, and they are interacting between them. So different colors are shown, are the hub genes, that is starting from the red and going to the yellow. So red one is the top one gene, and the yellow is the least interacting gene in top 10 gene. APOA1 gene have 54 other interactions, and LIPC gene have 32 other interactions. And in figure number 7, here we can see protein-protein interaction network, and cluster has been generated using MCL clustering. And here in this figure we can see there are two major clusters and two or three clusters which has only 2 or 3 genes, and it has very least significance. So the bigger cluster, it has 43 genes, and the main pathway that are involved in this cluster is high density lipoprotein particle pathway, and the second one, it has 14 genes, and the major pathway that are involved in this cluster is epoxy genase P450 pathway. So coming to the next analysis, which is gene enrichment analysis. So it has 4 major steps. First one is the biological processes, and in this biological processes we can see that plasma lipoprotein particle remodeling is one of the most significant here, but regulation of lipid localization has most number of gene involved in it. Reason being that the circle is showing the number of genes, and the distance is showing the significance. So coming to the cellular component enrichment analysis, we can here see that high density lipoprotein particle is most significant, but vessel remain is that which has most number of gene involved in it. So coming to the molecular function, arachidonic acid monooxidase activity is significantly shown as a functional level, as shown as a function, most significant molecular function. But so far the number of genes is concerned, tetrapylomole binding has most number of genes in it, involved in it. And so last but not the least, the cake pathway enrichment analysis. In this whole process, the most common pathways are arachidonic acid metabolism, cholesterol metabolism. These two metabolism are more or less same, because it has same number of genes and quite significant as well. So this much is for the RNA-seq data analysis. Now going to the mass spectrometry data analysis result. So in mass spectrometry data approach, 156 proteins were common, and 960 proteins were for Alzheimer, and 240 proteins were unique for Parkinson. and using 156 proteins I have generated the protein-protein interaction, which is shown in figure number 10, and the circle is representing the protein and the distance between them is showing the interaction, that is the edges. So now after seeing the protein-protein interaction, I try to identify the hub proteins, that is top 10 proteins. So PDHB, PDHA1, MDH2, VDAC1, CS, ATPF1A, ATP5F1B, TP1I1, HSPA9. These are the top 10 proteins, and here is the enrichment analysis shown in the right hand side of the slide. A is the biological process, B is the cellular component, C is the molecular function, and D is the cake pathway. So far in the proteomics approach, the most significant biological processes came out is the generation of precursor metabolites and energy, and though it is a significant one, and it has most number of genes involved in it, and the least number of genes has least significant. And so far we can see here in the cellular component, mitochondrial matrix is the most active cellular component of the other out of all other cellular components, but extracellular vesicles and extracellular exosomes have most number of proteins involved in it, almost 60 proteins involved in it. And in molecular function, NAD binding, small molecule binding has NAD binding is the significant one, but the number of proteins that is larger is small molecular binding and nucleotide binding. And so far the cake pathway is concerned, carbon metabolism is the significant one, and it has most number of protein involved in it. So this is the discussion part, and here I have mentioned the role of the protein, which I have already discussed in the result part. So this is the discussion part. I have already mentioned it in the result. So as coming to the conclusion, so neurodegenerative disorder is a condition where message from one neuron to another neuron get hindered by so many of the reasons. Few of them has been reported, and they are amyloid plaque formation, tangles, or alpha-synucleic in diseases like Alzheimer and Parkinson. My approach have advantages like it is cost saving, life risk is zero, and I did my work on already available data, and it is statistically significant. So I have identified top 10 genes that are common for Alzheimer and Parkinson's, and top 10 proteins that are common for Alzheimer and Parkinson's, that are associated with biological processes such as protein lipid complex, protein containing complex remodeling, plasma lipoprotein particles remodeling. So far our proteomics approach is concerned, it suggests protein that are involved in metabolic precursors, enzyme energies such as TCA cycles, pyruvate metabolism, and ADH metabolism processes, along with cellular respiration, and all are mainly affected pathways common to both the degenerative disorders. A synergy between proteomics and genomics approach leads to the identification of common possible metabolite points that converge to cause neurodegenerative disorders. The findings seems to imply common convergence point at molecular level influencing several cellular processes related to disorders, and can prove to be the key therapeutic targets as well as for drug companies and for further in vivo studies. So here, thanking to my professor Dr. Parvez Babbar and my biochemistry department team, Dr. Seema Kalra, Dr. Manisha Pandey, Dr. M. Abdul Karim, Dr. Arvind Kumar Shakya. And last not the least, here the quotation of the day: the science of today is the technology for tomorrow. Thank you so much.